Is it possible to regrow any limb or organ, even a whole head? Absolutely. Hey there, welcome to Science is You. If you're enjoying these videos, please subscribe to help this channel get off the ground and to support our mission of making real scientific papers enjoyable and accessible for everyone. Thanks for watching. Obviously, the capacity to regrow part of our brain would be of huge benefit to trauma patients or people with neurodegenerative diseases. Unbelievably, some animals have amazing regenerative capabilities and can even regrow their whole head, including their brain. One such animal, the planarian flatworm, looks very different from humans, but we share many of the same organ systems, such as a central nervous system with a brain and eyes. But what enables planarians to regenerate so incredibly well while humans can't? That's exactly what the authors of this research study set out to investigate. Planarian regeneration depends largely on a pool of cells called stem cells that are capable of turning into any cell in the body. This process, called differentiation, occurs largely through turning different genes on or off. But what tells the cells which genes to turn on or off at what time? Let's take a deeper look at what a gene really is. DNA, the genetic material, is sort of like an instruction manual describing how to build your cells. But if you imagine the instruction manual being stored in a compact form like a USB, then you have to download and unzip the folder, then use a computer to translate the digital data into a form we can read and understand. Similarly, DNA has to be copied and transcribed in order to be used as the blueprint to make proteins. These are molecular machines that enable cells to carry out their different functions. For example, the cells in your skin and the cells in your eye generally have the same DNA, but they're making different proteins, so they look and function very differently from one another. There are lots of different proteins in your cells, encoded for each by small sections of the DNA called genes, sort of like pages in the instruction manual describing the assembly of different components. But how do these genes get turned on and off? Often this is controlled by a transcription factor, which are specialized proteins that attach onto a gene and enable the copying and transcribing process to start. But then what signals cause the transcription factors to get produced? Clearly, this can get very complicated and lots of signaling pathways and feedback loops are possible. How one type of cell turns into another is one of the biggest and most frequently studied mysteries in molecular biology. So how can we examine this? Here, the authors wanted to see what genes are turned on or off during the process of planarian head regeneration. How can we observe this process in the lab? That part's simple, cut off their heads. But to look at what genes are turned on or being produced as the flatworms begin to regenerate, the authors looked at what RNA was present. You've probably heard of DNA sequencing, which tells us what DNA is present in the cell. Similarly, RNA sequencing tells us what RNA molecules have been transcribed from DNA and are now present in the cell, ready to make proteins. How can you get RNA out of the planarians, though? You might have tried the experiment where DNA is extracted from a strawberry by adding soap and other chemicals to break down and destroy the cells while leaving the DNA intact. Extracting RNA from the planarian is somewhat similar. Here the authors extracted the RNA from planarians either 12, 36, or 72 hours after cutting off their heads and compared them to healthy unharmed planarians. They found over 900 RNAs were increased by at least double in regenerating worms versus unharmed worms. Not surprisingly, among the genes with the highest production were those known to be produced in the brain, head, pharynx, and eyes. But there is a subset of genes with unknown functions. Could these be the genes that are responsible for triggering head regeneration? To see, the authors removed or inhibited each of these genes using RNA interference, or RNAi. In this procedure, small sections of RNA are fed to the worms where they enter the cells. Once inside the cells, the RNA pieces attach to one of the types of RNA that corresponds to a specific gene and prevents it from being further made into a protein. In this way, the function of individual genes can be evaluated by seeing how blocking their production into proteins affects the worms. So, if we block gene A from making protein A and the worm is no longer able to regenerate its head, then we know that gene A being active is necessary for head regeneration. Then maybe we could look for ways to sort of activate or turn on the equivalent of gene A in people. Basically, but of course things are more complicated than that, as many genes and their corresponding proteins may be involved in blocking the production of one might not completely block head regeneration. This is further complicated by the fact that turning on or off one gene like a transcription factor could affect the production of other genes. Also, with RNAi, genes are usually not completely blocked, just inhibited, since some RNA molecules might escape binding by the artificial RNA pieces fed to the worms and go on to form proteins. For example, the researchers here found 30 genes that caused a significant reduction in brain size of regenerated worms, with a severity range of 16 to 80%, including one gene that specifically caused defects in eye regeneration. In addition, the researchers found over 500 genes that were decreased in regenerating worms. These genes predominantly corresponded to cells of the intestines. To see where in the worm genes are turned on and producing RNA, the researchers used in situ hybridization, which is a technique that uses a molecule that glows a fluorescent color under the microscope that can attach to a specific RNA molecule, corresponding to its specific gene. The authors observed that the transcription factor SOX-B22 was produced in a small number of cells near the regenerating head, and blocking its production reduced brain size after regeneration and also reduced the animal's ability to respond to stimuli. Another notable gene is RUNT, which was needed for proper eye regeneration. Other genes caused decreased brain size through unknown mechanisms. Among the 30 genes identified were several involved in wound response, extracellular proteins, signaling molecules, brain structure, olfactory signaling, and in particular, several genes that encode secreted or membrane receptors that could function in signaling. A bonus finding from this study was the discovery of glial cells in the planarian brain. Glia are a diverse and essential pool of cells that are part of the mammalian nervous system that provide physical support to neurons and help maintain their environment. They're also called the glue of the brain. The discovery of glial cells in planarians makes them even more similar to us than previously thought. So how then can we regenerate parts of our head like eyes or damaged brain tissue? This study advanced our understanding of planarian regeneration and identified new cell types like glial cells that are present and involved. Many genes were uncovered that participate in regeneration, but the interactions between the protein products of these genes is complex, involving physical signals, chemical signals, and exact timing of molecular interactions. Still, although we still have a ways to go, with the similarities that exist between species, are we closer to regenerative abilities than we think? If you liked this video, don't forget to subscribe and remember,